Molecular evidence provides some of the most compelling proof for evolution. By examining DNA and proteins at the molecular level, scientists can directly compare the genetic makeup of different species. This approach reveals how closely related organisms are to one another based on their genetic similarities. The more similar the DNA and protein sequences between two species, the more recently they shared a common ancestor. This molecular approach gives us a quantifiable, objective way to trace evolutionary relationships across all life on Earth. Molecular evidence is crucial for three main reasons. First, it allows direct comparison of genetic material across different species, giving us an unambiguous view of their biological relationships. Second, it provides quantifiable measurements of evolutionary relationships. Unlike subjective observations, we can count the exact number of genetic differences between organisms. Third, molecular data reveals the timing and patterns of common ancestry. By analyzing mutation rates, scientists can estimate when different species diverged from their shared ancestors. This makes molecular evidence one of the most powerful tools in evolutionary biology. One of the most striking pieces of evidence for evolution is that all life on Earth uses the exact same genetic code. Every living organism, from bacteria to plants to animals, uses the same 4 DNA bases: adenine, thymine, guanine, and cytosine. This universal genetic language strongly suggests that all life descended from a common ancestor. If different forms of life had separate origins, we would expect them to use different genetic systems. But instead, all organisms share this fundamental molecular machinery, pointing to a shared evolutionary heritage that connects every living thing on our planet. When scientists compare DNA sequences between species, they look at specific genes and count the differences. In this example, we're comparing a short DNA sequence between humans and chimpanzees. Notice that both sequences are nearly identical, with the same nucleotide bases appearing in the same positions. This high degree of similarity tells us that humans and chimpanzees are very closely related evolutionarily. The principle is straightforward. More similar sequences indicate a closer evolutionary relationship. Species that diverged more recently will have accumulated fewer genetic differences, while those that split apart long ago will show more variations in their DNA. Looking at overall genetic similarity gives us a clear picture of evolutionary relationships. Humans share 98.8% of their DNA with chimpanzees, our closest living relatives. We share 85% with mice, which are fellow mammals but from a more distant branch. With chickens, we share 65%, reflecting our more distant relationship as vertebrates. Remarkably, we even share 50% of our genes with bananas, because we both inherited basic cellular machinery from ancient common ancestors. This pattern of decreasing similarity perfectly matches what evolutionary theory predicts. Closer relatives on the evolutionary tree share more DNA, while more distant relatives share less. Proteins provide another powerful line of molecular evidence. Certain proteins, like Cytochrome C, are found across all species because they perform essential functions. By comparing the amino acid sequences that make up these proteins, scientists can assess evolutionary relationships. Fewer differences in amino acid sequences indicate closer evolutionary ties. What's particularly compelling is that protein evidence independently confirms what DNA evidence shows. When scientists compare organisms using proteins, they arrive at the same evolutionary relationships as when using DNA. This convergence of multiple independent lines of evidence strongly supports evolutionary theory. Cytochrome C is a protein involved in cellular respiration that's found in nearly all organisms. By counting the amino acid differences in this protein between species, we can measure evolutionary distance. Humans and chimpanzees have 0 differences in their cytochrome C, confirming our extremely close relationship. Humans and rhesus monkeys differ by just 1 amino acid. Dogs differ by 11 amino acids, reflecting that we're more distantly related mammals. Finally, yeast differs by 56 amino acids, showing our vast evolutionary distance from fungi. This pattern perfectly mirrors the evolutionary tree constructed from other evidence, providing independent confirmation of species relationships. Pseudogenes are particularly fascinating pieces of molecular evidence. These are broken, non-functional DNA sequences that no longer code for working proteins. They're like molecular fossils within our genome. What makes pseudogenes so compelling is when the same gene is broken in the same way across multiple related species. For example, the gene for producing vitamin C is broken in humans and all other primates, but it works fine in most other mammals. This shared broken gene is best explained by common ancestry. All primates inherited this same genetic error from a common ancestor who first experienced the mutation. If species were separately created, there's no reason they would share identical broken genes. The molecular clock is a technique that uses mutation rates to estimate when species diverged. Mutations accumulate in DNA at relatively constant rates over time, like a ticking clock. By counting the number of genetic differences between two species and knowing the average mutation rate, scientists can estimate how long ago those species shared a common ancestor. More differences indicate a longer time since divergence. This molecular clock has been calibrated using fossil evidence and provides remarkably consistent estimates of evolutionary timing. It's become an invaluable tool for reconstructing the timeline of evolution and understanding when major evolutionary events occurred. Let's summarize the key points about molecular evidence. First, DNA and protein similarities directly demonstrate evolutionary relationships between organisms. Second, greater similarity indicates more recent common ancestry, giving us a clear measure of relatedness. Third, molecular evidence provides quantifiable, objective data for evolution that can be measured and tested. Finally, multiple independent lines of molecular evidence, including DNA sequences, protein sequences, pseudogenes, and molecular clocks, all converge on the same evolutionary conclusions. This convergence of evidence from different molecular sources provides overwhelming support for the theory of evolution and our understanding of life's interconnected history.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.